I think I should begin with an apology. I don't speak Spanish, so I have no option but to speak in English. But I'd also like to begin by thanking my friend Miguel and other organisers for giving me the opportunity to come and hear what you have to say today and to give this talk. My presentation is going to be largely historical in that the first reintroduction programme I was involved with started in the early 1970s. The world was very different then. We knew of no previous reintroduction programmes on raptors, not successful programmes anyway. So the methodology had not been developed. Also, we had no satellite tracking, so that when birds disappeared from our area, the release area, we'd no idea where they went, and we'd no idea what killed them or how they died or when they died. That sort of information we now have as a matter of routine. Well, just to remind you, reintroduction is sort of any attempt to re-establish a species in part of its natural range, an area from which it was eliminated in the past, but for which conditions still seem suitable. Usually, reintroductions involve native species that are unlikely to recolonize naturally, at least in reasonable time. But they've become reintroductions an increasingly important component of conservation practice, as you know. They don't replace any other conservation actions. We still need protective legislation. We still need species management and nature reserves, but reintroductions are just another tool in our conservation toolbox. As all of you are aware, the reintroduction is simply a restoration of a lost population. But in the process, of course, we are extending the range of the species, and in this way, we're probably giving it greater long-term security, because the larger the range of a species, the more secure it's likely to be in the long term. Sometimes reintroduction programs have been aimed at saving a doomed remnant population. Some populations live in areas where they could not survive because of change, and so we take them and we put them somewhere else where they're better able to survive. And a final thing about reintroductions, it's not a justification, it's just a consequence, is that there's very often great public engagement and promotion of tourism as people come in to see the new species in their area. I'll skip that one. Well, my own involvement in reintroduction programs are listed on the slide there. There are two in Britain, as you see, white-tailed eagle and red kite. And there are three in the United States, peregrine falcon, aplomado falcon, and California condor. Now, the two in Britain both involved getting young birds from other populations, from European populations, and releasing them in Britain, where the birds have become extinct. The three in the United States all involved the captive breeding of individuals and using captive bred young to reinstate the population. Well, turning to the first of these, you'll all know that the peregrine falcon was eliminated by DDT and other organochlorine pesticides from much of its range. In fact, it disappeared from the whole of the eastern part of the United States and low latitude Canada. And when there were no birds left in the eastern states, a group of falconers, 4 falconers, headed by this man, Tom Cade, who was an academic, they decided they would try for the first time to breed peregrines in captivity to produce a population for reintroduction to the wild, because the population in their area, the eastern states, was extinct already. Now, the only surviving falcons from the original population in eastern North America were all in the hands of falconers. These falconers had taken the birds as chicks, young chicks, and reared them artificially, and they were all imprinted on humans. And they regarded humans as sexual partners. They would not breed these birds with conspecifics. And by chance, one of the owners of these birds, a falconer, his bird was so much in love with him that every time he went in, it would copulate with him, but only when he was wearing that particular hat. And he hit upon the idea of collecting the semen from the hat in a syringe and using that semen that he could pick up to fertilize captive females. And he did that. And that was successful. The birds, some of the females laid eggs, and those eggs were incubated and raised artificially. So this was, as far as we know, the first peregrines that were ever bred in captivity. They were all fathered by this one male of rather strange habits. It was clearly not enough to establish a wild population. They had to get birds from elsewhere too. But the local population, as I mentioned, was extinct, so they had to get birds from other surviving populations, including in Britain. We sent them birds. And their idea was that they would use birds from a number of different areas, birds that looked like the original stock, interbreed them in captivity, and then release these hybrid offspring and let natural selection take its course to produce a better adapted population. They had no option but to work this way. And they needed to produce not just a few birds, but as large a number as possible, because the idea was to try and re-establish the bird over the whole of the United States and beyond. Now, initially, these birds were released from boxes that were placed on the old nesting cliffs. The birds were put in there when they were about half grown, and they had no contact with people. The food, you can see there are pipes going down into that cage, and when the birds were put in the cage, the food, they were fed by dropping food in from above. They never saw people, but once they were old enough to fly, the box was opened and they were allowed to fly out. And then food was placed in the surrounding area and they were hacked. But there was a problem, that most of the young released in this way were predated by great horned owls. That's the big bubo owl, rather like European eagle owl. And most of the young peregrines that had been painfully reared were eaten in that way. So they decided they'd had to put the young, they had to release the young in places where they were free of predation. And some of the main sites they used were buildings in city. Now, previously, there had only been one record in the whole of North America of peregrines nesting on a building, and this was in Montreal, and it was only one female that was involved. Once that female died, that was the end of that habit. No other peregrines were recorded as nesting in buildings. But in the birds that they released from buildings, some of them returned to cliffs to breed, the ancestral habitat, but others themselves started nesting on cliffs. And that was the start of a new habit which spread rapidly. And nowadays, you can find peregrines breeding in almost every city in North America, feeding on the local pigeons. Now, the pesticides that caused the decline were banned in America in the early 1970s, and in the next 20-year period, Cade and his team produced more than 5,000 young by this method, by captive breeding, and releasing them at different sites in different cities around the country. And now they're widespread, as you know, and commoner than they've ever recorded in the past. Now, the lessons from this program for me were, first of all, that you can breed these birds in captivity, but you need to be very careful of imprinting. You need to protect the young from predators. that the fourth lesson, it is possible to change the nesting traditions of a species to something that's more favourable in the prevailing environment. Now, we know that peregrines in Europe, in Spain particularly, had nested on buildings probably for centuries. But here they learnt that habit on their own without human help. Now, moving on to the second species I want to talk about, which is the red kite. We had a tiny remnant population left in Britain, in a western part of the country, in Wales. It was very small in numbers and highly inbred. And the population in the 1930s was known to number only 5 individuals, but they were carefully protected, and they gradually increased to only about a dozen pairs in the 1980s. At that time, I worked on them. And analysis of the genetics of those birds at the time suggested that this whole population was derived from a single female. So that genetically they were very restricted in diversity. Now, this kite program was costing a lot of money and it was getting nowhere. So we decided that a reintroduction program was worth trying, using birds from continental Europe. And the Spanish authorities and the Swedish authorities were kind enough to grant permission for us to use birds from Spain and Sweden in a reintroduction program. Now, one of the main problems that the original population faced, that we studied in Wales, was rainfall, in that it's a very wet area, and a lot of the young died in the nest during periods of heavy rain. So when we made a reintroduction, we decided we'd put the birds in 2 eastern areas. In Britain, there's a gradient in rainfall. It's very heavy in the west and very light in the east. So we put the new birds in areas with very little rain. And we used the usual hacking method. We got the young birds, reared them on in aviaries like this, and when they could fly, they were opened and released, and then food was placed in the surrounding area, and they gradually learned to feed themselves. And this bird's from Spain, and this bred 2 years after it was released. And that bred in an area where kites had been absent for 400 years or so. Now, the 2 first release areas, there was the one in southeast England. They were birds from Spain, and there was one in North Scotland. They were birds from Sweden. And in the top graphs shows the number of birds released each year. It was the same in both areas. And in both areas, the reproductive rate was high. They were producing lots of young. But as you can see, in the southern England population, that increased extremely rapidly, whereas the northern Scotland, despite producing lots of young, it increased very slowly. And research showed that this was a problem of adult mortality. People were still killing them in the interests of game preservation. That problem has now been resolved to a large extent, and that population has increased. Now, our original plan was to, as I mentioned, to release birds in 2 areas, southeast England, which is there, and northeast Scotland, which is there. And our original plan was to join these 2 areas together with a succession of other release sites. From the northern one, we moved gradually southwards, and from the southern one, we moved gradually northwards over a period of 15 years or so. And that is the original population in Wales. And that was, we thought, the quickest way to get the whole country recolonized. And that has worked really extremely well. The bird is now found over more or less the whole of Britain and is now breeding in every British county. And that was achieved within 20 years or so. The 2 things here, we benefited enormously from experience in early programs, so we knew what to do. and despite continuing human killing of some birds, they were very successful. And it showed us the benefit of having many different release areas rather than just one or two. That's the quickest way to get the bird to extend its range. Now, the last one, we had very heavy public engagement programs in these reintroductions. We used to invite school children to every release site to see the birds, handle the birds, and get interested in them. And of course, they pass that on to their parents. But the last case I want to give is the California condor, a carrion-eating bird, as you all know, equivalent to the Old World vultures. And the history of this bird is that it once spread widely over the southwestern United States, but declined through continual human action, mainly shooting. By 1900, the whole population was restricted to a small area in California, and by 1982, only 22 birds remained in the wild. This bird had never been bred in captivity. Well, that's a close-up of the bird. It's not the most attractive of birds, but it's very charming. Anyway, captive breeding seemed to be the only opposition because the population was declining so rapidly in the wild. But despite much opposition, the decision was taken to take the birds into breeding centres. And there were two breeding centres, one in California and the other one in Idaho, led by Tom Cade of peregrine fame. The birds were housed in pairs in big aviaries, and each aviary had a nest box, and it also had a weighing machine that the birds could be weighed from a distance so that you could check how they were doing. And otherwise, they had no contact with humanity. And luckily, the birds bred readily in captivity, or at least they readily laid eggs. Now, in the wild, a condor normally produces only one egg every 2 years. It takes so long to rear a chick that they can only nest once every second year. But in captivity, it was discovered, if you take the eggs as they're laid, you can get 2 eggs from each female in a year. So in 2 years, you get 4 eggs instead of 1 in the wild, eggs which you can incubate artificially. And that's a photograph of the incubation room at the Peregrine Fund headquarters in Idaho. Each of those incubators contains a single precious condor egg, and the incubation period lasts over 2 months. Once the young are hatched, they're placed on the left there, you can see those brooders. They keep the young warm, and the young can be fed through those, but they're not fed by people. They're fed by people, but with a puppet head. That goes over somebody's hand, and you can manipulate it at the end and feed the young bird. So that is the only contact they have. They never see a full human being. And it takes them more than 5 months to reach more or less full size. But when they're half grown, they're taken out of those areas and put in aviaries together with a number of other young condors once they can feed themselves. And then when they're about 5 months old, they're placed in these outside areas in the release area. And this release area is in the Grand Canyon in Arizona. And this area was chosen because it's a large national park. There is no shooting in the area. And the National Park Authority, it's a part of the original range of the condor. Anyway, after probably 2 months in those cages, they're about 3 meters tall, the birds were let out, and again, food was put in the surrounding area for them to feed. Well, all this went very well. The birds were tagged so that they could be read by members of the public through binoculars, but they also carried radio transmitters. 7 years after the first young were reduced, we got the first nest. And that's a picture of a bird that's taken from a long way away in a cave in the side of the Grand Canyon. And the interesting thing was that bird reared young, And when the climber got down to the nest site, he brought some material from the bottom of the cage up again. And in that arid environment, nothing ever rots. It just lasts forever. And in the old rubbish, at the bottom of that nest cave, there was some old condor eggshell that must have been at least 200 years old. So this demonstrated to us that that bird had chosen to nest in a traditional old condor nest site. It had chosen the same place as its ancestors did, and we found that very encouraging. So all that went well, and the population increased to more than, as you can see, more than 50 individuals. But it soon became clear that there was a big problem with mortality. A lot of the birds were found dead, and because they had radio transmitters, they could be recovered quickly. And it was soon found that the biggest problem was lead poisoning. These birds were feeding on animals that had been shot and left outside. They'd not been recovered. And condors were finding these dead animals and feeding on them and taking in lead bullets or lead shot. That's an X-ray showing lead pellets. That's not in the bird's muscles, that's in the digestive tract. So they were getting lead in that way. So the decision was taken to catch up all the, well, we did a survey in the winters of 2006 to 7, and the entire population was caught up. Blood samples were taken to measure their lead levels, and 90% of them showed levels of lead indicating exposure. And about 70% of those were given treatment. It's called chelation treatment. You feed the bird a particular chemical, and that changes the lead into a form that it can be excreted. So it gets rid of it from the body. That's the method that was used on domestic animals and on people suffering from lead poisoning. And it works on condors. But nevertheless, some died. Now, from this, the decision was taken to catch up or try and catch every single condor in the population every year, sample the blood, and give them this chelation therapy to try and reduce the problem of lead poisoning. Now, the problem of lead poisoning, there was no shooting in the national park, but the birds were ranging over such wide areas for food that they picked up this lead outside the park. Many of these birds would fly 100 kilometers or more just for a single meal and then come back again. So there was no way of preventing this lead poisoning. So a difficult decision was taken. It was obviously going to take a long time to get hunters to change from lead to some other less toxic ammunition. So the decision was taken to continue to breed the birds in captivity, to add to them every year another 10 or a dozen birds every year from captive breeding, and at the same time to continue with this therapy, this chelation treatment, which makes the program extremely expensive and also an ongoing expense. But so far, this is part of the chelation therapy when the birds are caught up. But we also had a problem with avian flu about 3 years ago when 5 or 6 condors all died together because of avian flu. So now whenever the birds are caught up for their therapy, the lead sampling, they're also vaccinated against avian flu. Well, this program so far has been funded mainly by private individuals who've put money into it, but also government money. But government money, of course, under the current president of the United States, that funding has been cut, so we're not quite sure what the future of this program will be. But anyway, that's where we stand at the moment. I want to end by just mentioning a few issues connected with reintroduction programs that we again can take for granted. I want to talk first of all about filipatry. That's the tendency of young animals to return to the area near where they were hatched or reared and breed there themselves. And these are some data, not from a large raptor, but a small one. That's the European sparrowhawk, Accipiter nisus, and this is from my study in the south of Scotland. Now, on this diagram, if you look at the middle of that diagram, represents the nesting area where the bird was raised. And all those dots show the positions in the surrounding area where these birds were found in a later breeding season. So you can see that most of these birds, young birds, had settled to nest within 10 or 20 kilometers of where they themselves were raised, but some had gone much further. And the lower part of that diagram gives the density of recoveries of recovered birds at different distances from the birthplace. And you can see it falls off exponentially. Now, that kind of pattern has been discovered, I think, in almost every bird species that's been examined, including raptors. But of course, the scale differs. Eagles would disperse much longer distances than these small sparrowhawks. But it is upon this filipatry that the success of all our reintroduction programs depends, because we depend on birds coming back around the area where they're released. If they didn't do that, we'd have no chance of success. They'd scatter over such wider areas. Now, I wanted also to say a little bit about genetic issues. The general program has been to choose birds for reintroduction that are as close as possible to the original population genetically. But we are dealing with a reduced gene pool in that we only introduce a few individuals, a small part of a population. So that reduces the gene pool, but also we're usually taking birds from a population that has already been reduced in the wild. So the gene pool, we can assume, is very much reduced on what it once was. But I've listed there 7 different species, is it 7 or 5 different species, all of which have given rise to viable populations from a small number of individuals. These all less than 10 individuals have subsequently been bred together to produce in captivity viable populations. Now, the most extreme example was this species, the Mauritius kestrel, which was reduced in the wild to just 4 individuals before they were taken into captivity and bred. And there was only one, I think one male in that population. But these individuals became rare because they nested on cliffs. It was a cliff nesting population. But they were destroyed by introduced monkeys, which lived on the cliffs, found the nest and destroyed the eggs or chicks. So the idea was, when these birds were released from captivity, they were released from boxes in trees. And when they returned to breed, they also nested in boxes. So it was another example of changing the nesting culture of a population to a different, safer site. And there are now between 300 and 400 Mauritius kestrels nesting in the wild, all in boxes in trees in a viable population. So even though you start with a very restricted gene pool, we do have examples of a thriving population being built up from that. If I have time, Alex, just mentioning the main lessons that I think we've learned on reintroductions on the past 50 years, including those programs undertaken here in Spain. We know that the methodology that we have works, and it will work well whether we use wild or captive individuals. It's much better to put out large numbers of individuals in a small number of years than to spread the introductions with a small number over a long period of years. That's because in birds like eagles that don't breed until they're 5 years old, if you only put them out a bit at a time, most of the birds are dead before there's enough around to breed. In captive breeding, artificial incubation and rearing, that's much more successful than leaving it to the parents, which often fail. With smaller species, protection from predators is crucial, and genetically found restricted birds, they've still produced viable populations. And multiple or many release sites is by far the quickest way to produce range expansion. And the seventh point, you can change the culture of nest site choice to get the birds to nest in safer sites. But releases have brought unexpected problems, the obvious ones being the condor. But they've also brought benefits, particularly in public engagement and tourism. So the long-term message is, I think, don't give up. Keep on trying, because even the most unlikely cases can be successful. Okay, thank you very much for listening.